Hello everybody. Today we're going to be talking about inclusion body disease in 2022 that affects boas and pythons, colubrids, corn snakes, vipers, python boas in general. And I want you to pay real close attention to this abstract. It is published on National Library of Medicine, the identification of Reptile viruses, Hardeman viruses, and novel chew virus in captive native Brazilian boa constrictors with void inclusion body disease. Now remember, this is when they already have the virus. When they already have it.
Boyd inclusion body disease, BIBD, is a transmissible viral disease of captive snakes that causes severe losses in snake collections worldwide. It is caused by Reptile infection, which can persist over several years without overt signs but is generally associated with the eventual death of the affected snakes. Thus far, reports have confirmed the existence of Reptile in captive snakes in North America, Europe, Asia, and Australia, but there is no evidence that it also occurs in wild snakes. BIBD affects boa species within the subfamily Boinian pythons in the family Pythonidae, the habitats of which do not naturally overlap. Here, we studied Brazilian captive snakes with BIBD using a metatranscriptomic approach, and we report the identification of novel Reptile Hartmanniviruses, and a new species in the family Chaviridae. The Reptile L segments identified are divergent enough to represent 6 novel species, while we found only a single novel Reptile S segment. Until now, Hartmanniviruses had been identified only in European captive boas with BIBD, and the present results increase the number of known Hartmanniviruses from 4 to 6. The newly identified chew virus showed 38.4%, 40.9%, and 48.1% amino acid identity to the nucleoprotein, glycoprotein, and RNA-dependent RNA polymerase, respectively, of its closest relative, Guangdong red-banded snake chew virus-like virus. Although we cannot rule out the possibility that the found viruses originated from imported snakes, the results suggest that the viruses could circulate in indigenous snake populations. Importance: Boyd inclusion body disease, BIBD, caused by Reptile infection, affects captive snake populations worldwide, but the reservoir hosts of Reptile remain unknown. Here, we report the identification of novel Reptile Hartmanniviruses, and a chew virus in captive Brazilian boas with BIBD. 3 of the 4 snakes studied showed co-infection with all 3 viruses, and one of the snakes harbored 3 novel Reptile L segments and 1 novel S segment. The samples originated from collections with Brazilian indigenous snakes only, which could indicate that these viruses circulate in wild snakes. The findings could further indicate that Boyd snakes are the natural reservoir of Reptile and Hartmanniviruses commonly found in captive snakes. The snakes infected with the novel chew virus all suffered from BIBD. It is therefore not possible to comment on its potential pathogenicity and contribution to the observed changes in the present case material.
So what they're saying here is these animals can be infected with multiple strains of different viruses caused by the weakening of the immune system due to the arenavirus. What people don't understand, these animals evolved with viruses. Humans evolved with viruses. You have viruses, millions of them in your body right now. They're all inactive. They can't detect them because of a phenomenon called viral dark matter. In other words, if the virus is not active, it can't be detected on the PCR tests. That doesn't mean you don't have them. Same thing with these animals. They're saying a natural reservoir. They're not sure where the arenavirus originates from. It's never been proven that rodents actually transferred it or other mammals transferred it over to these captive snakes. At least not yet. They haven't been able to prove it yet. But what they're saying is that's why they keep saying the original transmission remains unknown. They can't tell if the virus is part of their natural reservoir because of viral dark matter. They can't be detected. Just like in humans, they can see millions of viruses, or they think they're viruses, they're not sure. But because of the technology they currently are behind on, they just can't tell. They know they're there, they see them, but they're not doing anything. They have no idea what they do. And that's in humans. So you could just imagine snakes. Okay, these snakes been in the jungle for how many millions of years, and all these viruses have been around for how many millions of years. They evolved together. They're discovering like this new chew virus, they call it. They can be all infected simultaneously with every strain at the same time. Because what appears to be happening is once the arenavirus, at least, starts to weaken their immune system, it starts waking up the viruses that they already have. Okay? That's how they're getting infected with all these other viruses. And it's unclear if all these other strains are airborne or not. They very well could be. Is the chew virus airborne? Very possible. I don't know. But yeah, why not? But I'm not worried about airborne viruses. I'll show you why here in a minute. I'm going to share what I do, what I've been doing for a long time now to keep, we'll say, their viruses in remission. And that's basically as a human being what you do. You take vitamins and you take care of yourself, not to catch viruses, but to keep the viruses you have in your body that are part of your DNA, that are just a part of you, that makes you you, and makes a snake a snake. You keep those viruses in remission. That's what you're basically doing without really realizing it. So yes, every animal you see on YouTube, world-renowned breeders, I don't care who it is, morpho, captive snake, whatever, python, colubrid, they already have viruses in their natural reservoir, and that's what they call it. But they're not sure if the arenavirus is part of their natural reservoir. That still remains to be undetermined. So yes, in the wild, these animals can be pulled out of the jungle, be blood tested, every one of them. None of them are sick. They all pass the PCR tests, right? Everything's good. But then you ship them. They go through terrible conditions. They sit God knows where for how long before they get transported again. Now they're stressed out. They moved around to a pet store and then they moved to your place. And the next thing you know, you have the arenavirus or the chew virus or whatever virus you want to call it. That's how this goes. It's exactly how this goes. All the evidence is there pointing in that direction. It's viruses that they already have that are in remission in the wild. They stay in remission just like they do very well with their own parasites, but then you bring them into captivity. You have this problem, they need medical treatment immediately to stay alive, to be treated for these parasites, internal parasites, when in the wild they have no problems with them. Same thing with these viruses. They have no problems because of their temperatures, humidity, their natural environment. They evolved to their viruses where it doesn't bother them. In captivity, it's a different story. Just depending on the animal and how well they're taken care of, somebody could have a snake 25 years and not have a problem. That doesn't mean that there weren't viruses in there. Of course there is. The viruses that they evolved with are in that animal. They just were not the leading cause of death. It can take a couple months. It could take a year, 5 years, 10 years. Who knows? And the arenavirus, like I said, it starts out as this meningitis type infection like humans get, but then it starts unlocking these multiple old world viruses that they already have. And like I said, it's really going to be hard to tell, at least for the next couple of years, to know for sure if the arenavirus is actually just part of their natural reservoir. It's very possible. That is very possible. It could be the rodent's natural reservoir, just like bats. These boas eat bats in the wild. Bats are just loaded with viruses, deadly viruses. Terrible. Bats are probably the worst, and these boas eat them right up with no problem. Pythons too. Never bothers them. All right? But anyway, this is the light I use for them. This is great for reptile and amphibians that require low intensity levels. That's what this is. This is a low intensity UVB/UVA tropical light is what I use for each animal. 16 hours a day, 8 hours of darkness. Provides UVA and UVB for calcium absorption. Very important. Ideal for tropical frogs, snakes, and nocturnal geckos. Provides 6 to 8 microwatts of UVB at 12 inches. So this is very low level. That's why they get 16 hours a day. Because the viruses, what they do is they're going to block the vitamin C, vitamin D, all the receptors that they absorb vitamins naturally are going to be inhibited. This light constantly keeps the vitamin D flowing. Constantly. It's not going to raise the vitamin D levels, it's going to maintain the vitamin D levels instead of them dropping at dangerously low levels while they're infected. You want to constantly keep the vitamin D going. I used a hemp extract in my previous videos, as I showed you. It inhibits the viruses from taking full grip of the receptors. It does a wonderful job, in my opinion. I've had excellent luck with the way I've been doing things. And for disinfection, I'll show you the other bulb. Here's a 9-watt near UV light disinfecting. This is UVC. Very low spectrum. Very low. This is what you need. You don't want something overly powerful. They make lamps that can disinfect a room in 15 minutes with UVC. But it puts off a lot of ozone, and you have to be out of the room, and you have to take your animals out of the room. These are safe for you to be in the room and leave your animals in the room with it turned on. They're very low level, but it takes longer for them to do a good job. Average, the room I use it in is what? 12 foot, maybe 13 foot long by about 9 foot wide. Yeah, that's about right. 8 foot tall ceiling. No windows in that room whatsoever. And I leave this light turned on for 3 hours a day to disinfect the entire room every day. So basically what this light will do is it will kill any virus, airborne, whatever, laying on a countertop. This UVC light will completely wipe it out. That's what these are for. UVC lights are not to be exposed to your skin, powerful ones anyway. This near one's safe. They make another unit that's called FAR UVC. That's like a $1,600 system, but they're very effective. Even these bulbs. That system, I think you only have to leave on for 15 minutes, and it's safe for you to be in the room, and it disinfects the entire room within 15 minutes of any germs, bacteria that can cause viruses floating around in the air, whatever you want to call them. I call them germs, but people call them viruses, whatever. It just totally deactivates all the virus and germs. So you could have a snake room with a snake rack in it, turn this on 3 hours a day. There's no airborne pathogen going to get through. That's what these are designed for.
Okay, so what I do with Hercules here, he gets 16 hours a day, not just Hercules, all of them, of UVB/UVA light. 16 hours a day, 8 hours of darkness, constant vitamin D going, UVA, keep the feeding response going, treats his SAD condition, seasonal affective disorders that they can suffer from. And what I'll do is I'll take him out once a week, soak him for 3 hours. I'll unscrew the base up here where it's solid, because you don't want the bulb to shatter in your hand. I haven't had any problems with him climbing around it or anything, or any of them ever, so I have no issues with it. He just lays underneath it to soak up all the vitamins he's getting and the lighting that he's getting. But anyway, I'll take him out for 3 hours, and I'll screw in a separate UVC light, and I'll let the UVC decontaminate the tank while he's not in here once a week. And then I run UVC over here for 3 hours a day in the entire room to disinfect the entire room. 3 hours a day. These are very low level bulbs, safe to use with them in the room, but then they're in tanks, so it's not a problem. Same thing with the Aztec. And the same thing with the Guyana. All get low level UVB, UVA, and when they're not in their tanks, I replace the bulbs with UVC to decontaminate each tank, just to keep pathogens away that could ever possibly infect them, like anything airborne or anything like that. Very effective. That's what I do. That's how I keep the viruses contained. Once a week, UVC, and these guys get their natural lighting 16 hours a day, 8 hours of darkness. Same thing with Central American over there. Yep, that's how I do it. It's a big part in their cures and their treatments, that's for sure.